I can see that a couple more of participants have joined, and those of you who have just joined, hello everyone. We will be starting with the webinar in just a few more minutes. And meanwhile, we are waiting. It would be great if you could introduce yourself in the chat box. You can introduce yourself with your educational background and research interest. Few of the participants who joined initially, they have already introduced themselves that they are from zoology background, from bioinformatics backgrounds, from microbiology background. I'm curious to know about the rest of you guys as well. Please let me know in the chat. All right, so meanwhile, the participants are introducing themselves in the chat box. I'm just going to start with today's webinar. So hello, everyone. A very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for a hands-on webinar on Cheminformatics for Biomedical Drug Discovery Program. For those of you who are joining us for the very first time, the program is designed for beginners and students who are eager to learn about the fascinating field of computational structure biology and cheminformatics. And in today's webinar, we are joined by Dr. Puneet Kacker, mentor of the Cheminformatics program, who will cover topics on scientific search methods and organizing literature data as a part of beginning research work, explain what cheminformatics is and highlight the latest trends in the field, as well as answer some of the queries that the participants might be having of the program curriculum. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the Omics Logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And this training has been completed in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data. And as a part of the program, students will also have access to the Omics Logic resources, which takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path of the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. And this also comes with an access to the Omics Logic courses, which include courses on bioinformatics, precision medicine, multi-omics that makes use of genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Users will also be given directions to access the tBioInfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines, as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. And different stages of analysis are performed on different sections on this multi-omics platform. So now, to view the various courses and example projects, let us first sign up on the Omics Logic Learn portal. So for signing up, I'm sharing the link in the chat box for the participants. So please visit the link and I'll walk you through on how you can sign up, since I can see that a lot of the participants responded that they are joining the webinar for the very first time. So I'll walk you through a brief demo on how you can sign up. So for those participants who have just joined the webinar, please click on the create an account option that you see here. And to create an account, you can enter a name, email ID, and a password. Or you also have the option to sign up using any of your social media accounts. This could be your Google, Facebook account, Apple account, GitHub account, or even Twitter. And also, few of the participants have already joined the previous webinar. So for those, they can just simply click on the login now option and sign on the portal. So I'll pause for a couple of seconds for the participants to sign up. Once you have signed up, please put a yes in the chat box. And those of you who already have an account on the portal, please put a done so that I know that everyone has signed up. And once you have done, I'll walk you through the various courses and example projects that are there on the portal. Okay, Ruhul says done. What about rest of you guys? If you're facing any technical issues, you can let us know in the chat box as well, and our team will be happy to guide you. For those who have signed up, please put a yes in the chat box so that I can proceed further. Okay, perfect. I can see that participants are signing up. Thank you so much for your responses. All right, so meanwhile, I'm just going to sign in so the participants can sign up along. So for those of you who have just signed up, you can see that the very first interface that you see once you sign up is this various display of the courses that are there. So once you scroll down, you will see a search bar that says looking for a specific course. So in this search bar, you can enter the keyword that you're interested. So let's say you're interested in cheminformatics. So that will show you the courses that are related to the cheminformatics. And you can also browse through and navigate various other keywords and look at the various courses that are there on the portal based on the keyword. So let us say you're interested in machine learning, you can simply enter the keyword and that will show you the various courses and example projects that makes use of machine learning. So you can navigate through and explore at your own pace. Now, we have to set up your account as well. So for that, just click on the tab that says welcome back with your email ID. So once you click on this, you'll be able to see your account settings. Here, when you go to the icon that says profile, you'll be able to edit all of the options. So one of the very first important features that you need to have update is your profile image. So for that, you can simply click on the select file to upload and upload an image from your local system. And you also need to make sure that your name has to be in proper capitalization. And also you have the option to link your social media handles. So in my case, I have linked my LinkedIn account and my Org ID. And finally, another last important feature that you need to update is your bio. So here in my bio, I have mentioned my educational background and my research interests. So the participants can set up your profile similarly. And you will also see the various tabs that are there on the portal. So under the activity tab, the program mentor and the community manager will be tracking your learning progress. And under the courses tab, the courses that you are currently completing will be displayed. Finally, after the completion of a project, and once it is published on the portal, your project will be displayed under the projects tab. And once you enroll for any of the programs, for instance, the Cheminformatics program, you'll be able to track your program progress under the Programs tab. Finally, after the completion of your coursework and required assignments, you'll be able to receive the certificates under the Certificates tab. So that was all about navigating through the portal. So let me see. No one has any queries, and I can see that a lot of them have signed up. So if you are facing any technical issues or have any queries, please feel free to share it in the chat. But for now, let us move on to the next hands-on session for today's webinar. For that, I would like to pass on the stage to the program mentor, Dr. Puneet Kacker, who is a drug discovery scientist and industry analytics manager at Accenture. So over to you, sir.
Thank you very much, Sonalika. I just wanted to check if you are able to hear me properly.
Yes, sir, you're perfectly audible.
Okay, makes sense. Thank you very much. Hello, everyone. So let me share my screen. I have a small presentation, but a significant amount of hands-on, and we can discuss this call. Let me show my presentation here. Is there? Let me share my screen and it should be coming to you now. This screen. Yes. Okay. Let me know if you are able to see my screen.
Yes, we can see your screen.
Perfect. This is very good. Thank you. So let me connect this to just wanted to see how we can make this one down. I don't know. Uh, okay. I just wanted to bring the laser here, but I'm seeing this screen. But anyway, just don't worry about it. So today's session will be sorry. Today's session will be based on the research that we begin with the search, important aspects of any research. And I believe since you are attending this specific workshop and webinar, you are somewhere have a scientific curiosity, you have some hypothesis in mind, you have a great interest to learn new things in the area of life sciences, cheminformatics, chemistry, or maybe in a similar allied areas as well. So whenever you are starting or beginning your journey in in this area of research, the first and the foremost thing that is required to do hands-on is to search, extensively search repeatedly, search again and again, search every day, search to make your knowledge up to date, search to have your self acquainted well enough with the latest trends in technology, search sufficiently so that you understand whatever is already has been done in the field of your interest or not. So begin your research with the search is something which is very beginning of your assignment that you should always be comfortable with. But unfortunately, many times when we begin our journey of research at a very late stage, we understand how to do a better search, how we can manage our search results. What are the key aspects we should always consider while we are searching our research, our topic? What is the best ideal way? What is the best scenario? um, what is the right set of tools and technique we should use? What is the right search engine we should use? What is the right database we should search and rely on? How we can get the PDFs, how we can get the full text? There's so many things around just a search, and search being so important is a very much neglected in the initial phase, and that's why it causes a lot of problems to the research students. This session is dedicated some part of the search, how you can make your research journey more comfortable, more confident, more streamlined, more manageable in in your area with the right set of knowledge in place. So I have a very small presentation which will cover a couple of things. One is why search is so important. Second, we'll discuss about what are the two major key platforms or highly used platforms for biomedical literature search. When I say biomedical, it doesn't mean that the chemistry is not involved. Chemistry for biology is also involved here. Drug discovery is for the life sciences. It's connected closely. So you will find those articles also on those two platforms. Though they are very well known to you, but there are many interesting things you should always consider while searching or using these two platforms. Manage search results. You can get the search very easily. But it's not just a search or the keyword that you should use. It's the story begins once you get the results. If you get thousands of articles as a search results, how many would you read? How would you bucket them into different categories? How you can focus yourself so that a small amount of yet very powerful article should not miss from your eyes? How you can manage those things? So managing the search results is equally, and I believe is more important than just search other things. Maybe you have 5 different articles that are so important for your knowledge to complete your PhD work, or maybe a small project work, very, very fundamental articles. But how would you land up to those, and how would you keep those articles, ensuring that during you should read them all, you should flag them sufficiently at the right place, so that whenever you need them you can get those articles, and you can ensure that you can cite them in your thesis. Performing a systematic reviews, it's a different area, but it's not very well known to people, and especially in the clinical trials data. If you are looking for or doing for a systematic review, a review of the clinical trial studies results that have been done in the past, what is the right method to do that? It's not just collecting bunch of article and just start reporting them. No, it's a different method. It's a different way of accumulating the research in a way that it should only make a lot of sense in the final conclusion that you made. So it's just not citing the people, citing the article and telling their opinion. You need to come up your opinion as well in the reviews article. It's like you are reading a lot of articles and then saying, okay, our understanding is like this. This is what we observe. This is how our point of view should, this is how we think our point of view is like this. Right? Some conclusion has to be made. And you can make conclusion only if you are comparing the studies fairly in an unbiased fashion, right? So performing a systematic reviews, and certainly some key considerations that you should always keep in mind, may be very useful to you in the whole journey of your research. Let me know if you have any questions so far. If you have some questions, keep writing on the chat so that at the end we can take them. But I think we can start for the interesting things. Why search is so important? It's you are doing research not just because you need a degree. The major or more important aspect of the research is you want to contribute something. Whatever you are taking as a knowledge from literature search is somebody else's contribution in that field. That is, if you are doing a research, it is your accountable. You are accountable, and in a very honest and fair fashion, you should feel yourself responsible that you need to contribute in the enhancement and enrichment of the scientific research. It means that your output of the research should be such that it should progress the knowledge, it should enhance the knowledge of scientific research. It should have some contribution, no matter big or small, but it should not be duplicated. It should not it should not replicate the same stuff in a different wordings. Possibly that is also part of research. Maybe people who have reported so far for a topic which is very difficult for other people to understand, and your research is to make them simple. so that everybody can understand. That's also fine, but that's a big contribution. That's a great contribution, I believe. So consider that whatever you are doing in your research, you are the owner of that work, and you deliver that work in the most effective way. That's your responsibility, right? So search will help you to do that. How? You will search the things which are already done in the past. You will ensure that what you are thinking to do in your research has not been done so far, or maybe has not done that way yet. It means that you need to design your study in a way that it should further add on the information which has already been done. You take that information, bring your experiments in picture, and then bring the knowledge, add on the knowledge. Hmm? Point number two, it helps in formulating a research question and planning the study. When you are reviewing the literature which has been done in the past extensively, you are extensively searching those things. It will also help you to formulate what has been already done, and that should not be repeated in your experiment. Or if it is repeated, there should be a benchmark that should meet the criteria, which should be as per at par with the scientific guidelines. So whenever you are planning something, you need to ensure that this exact thing should not be done earlier. And this is possible once you keep on learning and reading what has been already done in the past. And that is feasible once you start doing a thorough searching, extensive searching. There should not be anything which is unturned so far, so that you can ensure that whatever you are thinking is in the right direction, meets the scientific requirements, and there is a need in the scientific domain that this knowledge has to come into the picture, right? Point number three, it improves the quality of research because you are adding a new knowledge, and in the way that has to be accepted by the scientific community, means your protocols. It improves the quality of research. If it improves the quality of research, you get the benefit of it, right? You feel more confident about it. Your research gets appreciated around the world. People start citing your articles, citing your publications. Scientific people invite you for the talks and for the discussions. You also possibly file the patents and move your output or scientific outcomes to a product. A product can help the mankind. It can improve the patient's life. So there are so many things when you genuinely do the research work, right? It is possible only if you meet all the criteria of um, scientific research, search, reading, reviewing, understanding the domain, formulating the right questions, putting down your hypothesis, doing the right set of experiments, generating the results which are acceptable to the scientific community, adopting the right set of protocols. It's like a very well connected things. So it improves the quality of research. Point number four, it significantly enhances your subject matter expertise. So if you start reading the articles uh, from the from the previous authors who have been doing that pioneer work in that area, you will certainly uh, become a subject matter expert, because you also have a very in-depth understanding of your work. So that also pays at a later stage in your career, and people recognize you because of your knowledge in that area. So it will help you. Point number five introduces you with the latest trends and technique. That's important. You may be thinking to design your experiment in a very traditional way, or maybe because you have studied that protocol method in your graduation or post graduation program. But when you are doing a research for the in the next 5 years time, with the you need to ensure that the right trends and techniques in that area that can really speed up, accelerate your findings with a more confidence, and whatever you were thinking to do in a 3 years, 5 years time, you can actually do in just 1 year time with more accuracy. For example, prediction of a three-dimensional structure of the protein. You were thinking you will do some MD simulations, you will do some homology based prediction, ab initio based predictions, and do that. But since machine learning has come into picture, maybe you find that whatever you are thinking to do by yourself, it's already there in the databases, and it is wide accepted. Maybe you can use that one, no need to build from scratch, and do the further analysis of maybe designing new molecules for the active site which were not previously in the purview of your research interest or research work periphery. So once you understand the latest tools and techniques and trends, it helps you to redefine your area into the research, and you can really run fast, more accurate, um, more productive, more efficient, for the area of your interest. So keep in mind that these things are possible once you do a thorough due diligence in the search. Important point is that whenever you search for something, you find hundreds of articles, maybe thousands of articles. Now it's not possible for a human being to read those thousands of articles in a short amount of time, because once you complete 1,000, by the time there are another set of 1,000 comes into the library. It is important that you need certain smart methods to your niche area, that you can effectively read them, understand them in a short amount of time, and you become expert in that area sooner as possible. So you need the right set of tools and techniques. I hope you understand why search is so important, and you will certainly recognize this as an important aspect whenever you do any begin any research work assigned to you in your program. Keep writing the questions if you have in mind. Maybe we'll come back and check those. Let me go to the second one. There are two very key and important platforms. There could be other proprietary, I would say, platform as well, but to me, publicly available, these two are the best one for biomedical literature search. One is PubMed, and second is Google Scholar. Where PubMed is highly focused on biomedical literature search, on the other side, the Google Scholar is something which can search anything. If it is a mathematics paper, if it is a applied mathematics on chemical sciences, biological sciences, if you are looking at the psychology paper, which is done on any behavioral studies on your disease area or so, no matter what it is, Google Scholar is much wider. Google Scholar also provides search results from accessory articles and um, reviewed pages from websites. For example, white pages. maybe from literature, which are patent documents, right? Those articles are also come into Google Scholar with the mention of the number of citations there. PubMed, on the other hand, has a very systematic, controlled index articles which provide some historical trends on the term results, their number of outputs, the quantity, the hits are very kind of accurate in that sense. And you can filter the article by reviews, by year, and so on. Google Scholar um, is something which uses a parent Google Google search, which is absolutely the great, absolutely great. So therefore, the search quality, you can say, the ontology of the search is really, really good. But nonetheless, both are. I use both of them. I recommend both of them because um, you can find certain differences in the outcome of the search results. PubMed and PubMed Central, another site on the NCBI, is in the PubMed Central is dedicated for the full text articles. Um, Google Scholar has the option that if the article is available in any PDF form anywhere on the search results, so that's added advantage there. However, PubMed does not link all the possible ways where, for example, university website where the article is available free of cost. So that's a limitation on the site. Another limitation from PubMed is the number of citations for the article. So it becomes little biased in a way that you pick always the article with the high citations. We'll come to that point, but not always true that the highly cited one is the best one. So maybe I can go quickly to show both of the search results from Google Scholar and PubMed. So I will open the Edge here. So I have this um, um, I have this one PubMed available here. So in case I wanted to see the results for say uh, Alzheimer's disease. So it also provides you the suggestions, which is fine. Alzheimer's disease, you get the results. You see how many results are there? 40,000 plus results are there. Well, it's very difficult for someone to read 40,000 article in a for a small site. But on the left hand side, you see a couple of filters option. The statistics says Alzheimer's disease. You can see then initially started around 1975 articles has started coming in, and they are rapidly increasing or consistently increasing year on year till 2023. We have quite a significant number of articles. Um, Other than that, you can filter them by abstract, full text, free full text, some associated supplementary data, whether they are book, clinical trial data reported, meta-analysis reported, some clinical trial data is reported, etc. Also, you can see from last 1 year, last 5 years, 10 years data is reported. Other options include the save the search results. For example, I will show you why this is so important, but saving is important. Email is important. You want to send these research results somewhere um, or save it in a clipboard, my bibliography collections also you can do. Another important thing is you can create alert, which is important. For example, I want to create alert and I can choose the frequency, day, format, etc. You can save it. I will show you when this is very important. It's an important point of discussion in my slide. And you can also have a display option where you can say, I need a summary, I need a abstract base um, show me abstract based things. So for example, article title, authors, affiliations, IDs, and then you have the abstract reported, etc., etc. Other informations are also reported here. So this is the a general description of the PubMed based search. You can narrow down the search, for example, adding more and more terms. For example, Alzheimer disease biomarkers. So initially we were having 40,000 articles. Now we will have 4,000 articles, just 10% of it. And then we can say machine learning, for example. Machine learning. I wanted to know the articles which have machine learning term, biomarker term, Alzheimer disease term. And see, there are 65 articles. So as you increases the area, focused area, it becomes reducing the number of articles. And whether they are making sense or not, you see Alzheimer disease drug development pipeline, and somewhere they have mentioned about in the articles. So it means that whenever you are searching like this, it is looking all over the article place where these terms are present. If you see Alzheimer applied machine learning, Alzheimer disease research, omics imaging and clinical data, which is also fine. So if you want to do the same search with Google Scholar, so let me go to the Scholar. Here's the Google Scholar. Okay, and I can search it here. So you see with this term, we have 27,000 results. And you see the PDF options are also given somewhere, wherever the article is available. You also see the difference that cited by is given, number of times this article has been cited. So it's a good merit to see whether the article is sufficiently read by the others and cited by others. Is one of the criteria of selecting the articles for your quick, quick review and search. You have the filter by this, and you say by default it's just not included or patents are excluded in the search. If you include the patents, then this 27,000 will become 28,000 or around 29,000 results. So that's little bit of difference here, but very fundamental difference. Of course, the top 10 articles here may not be top 10, certainly not the top 10 articles there, because the criteria of search is completely different, I believe. But I may be wrong here. For some articles, maybe you find both the places at the top 10 or so. But huge difference, right? 65 articles and you see 27,500 articles. And it becomes even more focused. That's a good thing. You need less article, but high quality articles. If you do with the advanced search, when you go to the advanced search, you say, I need machine learn. All the terms should be written. All the terms should be written in the title. Let me see if it produces any results or not. Let me see. OPQRS title. Let me see if it produces any result. 16 articles. So machine learning, Alzheimer disease. I believe biomarker is taking title and abstract. So you have biomarker, disease, Alzheimer disease, machine learning is there in the title. So this is how you can really focus on the so what I can do, I can download this 16 articles in a very specific format called save all results, and I will say PubMed method. So PubMed output file will be created for all the 16 results. Let me do this. I am doing intentionally, will tell you why, but let me go to the presentation again. All right. So this is good. PubMed, Google Scholar, there are certain differences. Both are good ways to search. and interesting. Now you get the results. What will you do with those results? Will you save those results somewhere in the in your hard drive? Um, if you are saving those results of PDF in some folders, how would you um, how would you recall them whenever you need them while writing the publication? Believe me, it's not that simple. You will really scratch your head if you are doing the literature management that way. Folders and subfolders, which article, how you rate, how you identify which articles to cite where. Every time, if you are doing this way of management, every time you need to write a single paragraph, you will search 10 times for the articles to cite. It's not absolutely a good way, a best way, the right way to manage your search results for the literature search. So, because there was a problem, there was a solution also created for it. People have created some specialized software for bibliographic management, and these are called kind of reference management or reference manager softwares. Some are, I would say many of them are commercially available. Some of them are freely available, but with a very limited, um, I would say, capacity. You can only search 19, manage 99 articles or so much of article. You need this much. You need some amount to pay after that, on so many terms and conditions. And they are little bit expensive softwares for a student to afford. On the other hand, with the God grace, we have a software which is freely available, open source, and does exactly the same right job. Here we are presenting two very well-known softwares in these two disciplines, two different uh, layers. One is EndNote, highly used, very powerful, but paid. On the right hand side, you have a Zotero, which is freely available. Reference manager does exactly the same job in a very efficient way, completely free of cost. If you're using a cloud, you may need to pay for after certain amount of storage that is required, but to some extent it is free. Standalone version is free. So I will come to that point. I do not have the EndNote with me, but Zotero I have installed for this specific presentation. And I will show you how you can manage your search results. Let me go there. So I installed Zotero by going to the website, Zotero. If you type Zotero, you will see your personal research assistant. Zotero is free, open source reference management software. Very good. You have very bold and clear line. Easy tool, helps to organize, annotate, cite, share research. You can download it. Available for Mac, Windows, Linux, and iOS. This is great. A very user-friendly software. No limitations on the standalone version. Can be connected to cloud. Uh, no worries about the data loss, etc. So it's very good. So I have this Zotero in place, and what I will do, I will go to, let me, yeah, let me go to this one. So what you see here on the left hand side is a hierarchy of certain options that how you can manage your library. You can rename them, you can do a lot of stuff here, folder and folders, subfolders, but they are visible to you in a tree-like fashion. You have on top is a menu buttons and some quick um, options. On the right hand side is called the information section about article that you have selected in the center pane. The center pane is just provides a list of the titles and associated information that you want to see. You can customize to that some extent, and you can do that. What you need to do, you can need to bring the articles into this platform. Now, bringing the articles can be done in multiple ways. One is either you can create manually. Second is you can certain second thing is you can search the results using PubMed or Google Scholar and save that file, and you can bring it here. And third thing is you can use the plugin which is also available with this Edge. I believe that's available here. Some plugin option that I see Zotero connector is available, so that can be also be used. So what is the most preferred way that I'm going to show it to you? The way I have done the search for Alzheimer's disease, biomarkers, and machine learning, what I did is basically I saved the results, all the results, how many, 19 articles in a PubMed format, and save the files. So I will go to the Zotero. I will say, let me delete this one, delete collection and items so that we can have a fresh. Now with this one, I will go to file, import, and I will say I need to import this file from my download, which is PubMed Alzheimer's set. And let me copy files to Zotero storage folder, which is fine. And it is importing. You see all the articles are now available. Now 16 articles are available here. How many were there? 16 articles were there as well. Not 19, but 16. So I have the 16 articles here with the titles reported here. And in the information pane, you can see a lot of information given, all the authors, abstract, etc. with volume, issues, page number, this and this is this, this is this. If you want to add some of the information that you have, you can always do that. It will be saved here. And since I connected Zotero with the free storage cloud that Zotero provides, it is syncing my library here. Now what I want is, since this is for machine learning and biomarker, I can rename this.
Sorry to interrupt you, sir. The screen is not moving.
It's not moving. Okay.
No, sir.
Let me reshare it. Okay.
We were able to see until you were showing the 16 results.
Okay, let me. Are you able to see it now?
Yes, sir. We can see a list of the research papers.
With the Zotero?
Yes, sir. In the Zotero, sir.
Okay, sorry. So I was saying this Zotero is a platform where you can see this hierarchical arrangement of the list of items. You have menu buttons, you have the toolbar options, you have the information pane here. In the center, you will see the list. So what I did, I can do the same thing here. I can delete the collection in the item again. Go to the file, import. I believe you can see all these things now. And I have downloaded this file and doing this one, copy to the Zotero and next. So this finishes my import of all the articles here. You see this is syncing to my Zotero drive, which is provided free of cost to me. Now, since these articles are very much of the machine learning, side, I would say machine learning markers. Okay. I can rename them. I can drag and drop it here so that they become the part one assignment. This is how I do that. This is a hierarchy. Same way you can do a lot of other folders creation. For example, here you can do the folder creation. You can say for cancer, and you can have the cancer. Let's like example, you have cancer and different type of sub cancer. For example, I can say new sub collection. Say cancer. So cancer within pancreatic cancer. You can have again new sub collections. That is all possible and very manageable. Now, if I go to the machine learning articles, if I click on any of the articles, I see a lot of information about the journal articles types, the abstract, the authors, the other information. So in case you have other information, you can certainly edit it, keep it saved, and it will automatically save. Each article is also tagged with some of the um, keywords, I would say tags for that, but just in case you want to add a new tag, you can go to the tags and add a new tag. Okay. For example, if this article is important to you and you want to say that a must read article, you can say, you can add a note here saying that, okay, notice here is important to cite in thesis. And you can just do this and this is all done. Now, if you go to the other section of this, so this is available here. You can always do information tags. And if you want to do some related article, that also you can search the related article and make the tag. Another important aspect is that I want to know what are the free articles available here. So what I will do, I will just do Control A here to select everything, right click here and find available PDFs. So very cool feature. It will try its best to see if the full texts are available and just not available. It will also add the PDF right after your article. Isn't a cool thing? I think it's a very cool thing, I believe. It's not that easy. It takes a lot of pain to get all those things in just few clicks. So it is searching, making and identifying articles wherever the full PDF is available. Um, and you will get it right there. Another cool feature here is you can double click on this one and go to that article directly, just in case you want to read more about it and see more of the information, just in case you want it. That is already there, right? So 27 euros article, expensive one. Okay, so that's another cool feature of Zotero, and it really helps you. Another point of using this one is not just managing this library, but also to cite article while you are writing your manuscript. Now that's a very important aspect. For this, how would you do that citation? Let me show it to you. I will go to MS Word. Let me see if I have the MS Word. Yes. This is the MS Word. Okay. And I want to cite some article. So you see, once you install the Zotero, you will see the Zotero option available in Microsoft Word by default. Now what you need to do is, you want to write a review article and saying some lines are like that. Machine learning significantly contributed in the discovery or in the identification of biomarkers, or say novel biomarkers for disease. For example, this is one of your lines in the article, and you are writing for, say, Journal of Medicinal Chemistry or so. So what you need to do is do this click here, add edit citation, and look for your Journal of Medicinal Chemistry. For example, here, it was not there by default. I added with the manage styles. You go to the manage style and look for the articles from there, Zotero side, and you can add this one here. Okay. So once you add it, you will see that is available. Now, if I go to the add text here, I can go to Journal of Medicinal Chemistry, GMAT Chem. I will say, okay, I want to cite an article. As soon as you just click add, the Zotero option will available. Can someone confirm me if you are able to see my screen with this Zotero search option on the Microsoft Word?
Yes, sir. We can see a red color rectangular box.
Yes, perfect. Now what I will do, I will search for my library. Everything is already there. So I will search for, let me see if there is novel biomarker. No, this biomarker is there. Okay, for example, I want to cite from the biomarker. Okay, say omics based. This one I want to cite. So this one is I have added. I just wanted to add one article. If you want to add, again, you can start searching it. For example, like this biomarker, you can add the another article, but I am not doing it this moment. And I just click enter here. You see, one is being added, but where is the article? So if you want to add the article, you just say add bibliography. So your first article has been added exactly in the format of what Journal of Medicinal Chemistry accepts in the publications. There's one thing. If you want to change the document in a different way, you just click on the different method, document preferences, select a different method, and you are just okay. You just need to say okay, and that's it. Everything will be done for you. So um, um, this is how you can cite your article in your manuscript, and you can keep your manuscript always ready to change to a particular journal guidelines. Okay, this is how you can save it. That's fine. You know everything. So this is how Zotero works briefly, and I think the search is also completed. You see 1, 2, 3, 4, 5, 6, 7 article out of 16 are available in PDF form. You can always go and look at the PDF. Very cool. You have the PDF article. I wish I knew this during my PhD time, but I think some of these cool features were already there. I use them very effectively, but some features are really, really cool. So that's briefly about the Zotero and managing your library. You can do a lot of bifurcation. For example, if there is a deep learning, let me know. No deep learning. There is no, there, for example, is there any diagnosis? So if there is a diagnosis, if there is a diagnosis, two articles are there for diagnosis. What I can do, I can create a subfolders in Alzheimer, machine learning, and say, and I can create a diagnosis. these two articles, I want to move them or just copy them into diagnosis. I will have the diagnosis 2 articles and this remain intact, 16 items. So just in case you have multiple articles, you want to remove duplicates within the same, then you can certainly do the duplicate removal, and this way you can do a lot of good stuff. Another thing is, just in case you want to add an article by the PubMed ID, for example, okay, info, let me go to the info of this article and see if there is a PubMed ID. So this is a PubMed ID, let me see. And the article name is Ageomics. Let me go and search. Let's see if this works. I don't know whether it takes the. Oh, it seems like it has already added Ageomics biomarker 2 times now. And this is already added now. And let me see if this is already done. Let me see if there is any. See, the duplicate article has come here, and you can now see both the articles here, right? So it is saying merge 2 items because it has found that these are 2 articles which are duplicated by some combination of title, IDs, etc. You can say, yes, merge 2 articles. Now you don't have any duplicate article, and this one will be only one time reported here. Okay, very cool. I believe you understand what I'm saying, but this is how it is. Good stuff. So on that part, we have covered the EndNote and Zotero part. Now coming to the systematic reviews. When you are doing a review of the specifically clinical trial data or significantly a similar, potentially a similar kind of data reported in by means of literature, there are methods to do systematic reviews. It's just not like finding last 5 years article and basis their full text availability, you are just doing the compilation and reporting it. So it has a very systematic ways of accumulating the data, excluding the articles, and then reporting it. It's called Prism A format. And the link for this publication is given there. I highly encourage you, if you are in the process of doing this or you have some plans of doing a systematic review, look into this one. The original article image is like this, which says identification, screening, eligibility, what is included. And generally it is how generally people are reporting this one in a reformatted way, but the ideology is exactly same. The method is exactly same. So they say that how many articles you found in the database. Is there any additional source of data? You combine the articles. These many articles were there after removing the duplicates. Now you have screened the article basis the requirements. So you screen all the articles and you have excluded the articles because they were not meeting the criteria. Now eligibility is whether the description required for the review is matching the requirement. What are the articles are required? So you are saying you are excluding some of the articles here, 103, and the breakup is also given because there was no clear qualitative component. Let me zoom in if I possible. Uh, I don't know how to zoom it here. Early phase screening prevention in ineligible RCT, random clinical trial. Population, ineligible phenomena of interest, abstract only, duplicate. So all those things will be excluded. Whatever is remaining for qualitative research, you study 45 articles and you have made conclusion on all 45 articles. So this is how you have to present your workflow, and then you do a meta-analysis called quantitative synthesis of the search and the analysis. Now coming to the last section, which are set of key consideration. I think we are on the top of the hour, but I will take just another minute to complete it. That uh, s- while searching the article, it's important that citations is one of the key criteria, but not always, because there are some great articles maybe unnoticed somehow because of the lack of right keyword, or maybe availability in the full text, or maybe somebody just not recognize the merit of that article, right? Um, and also maybe article is very new to be cited heavily. So those things are considerable when you are searching the articles and selecting the articles for your references. You can prefer articles with good illustration in simple English so that you can understand them fully. You can imagine what they are saying, sometimes difficult, and illustration really helps a lot there. So you can do a create alert in Google and PubMed. Use that functionality is very important because it will keeps you up to date with the latest recent articles coming or indexed in those platforms. So you will understand them what is the latest going on there. You can utilize cloud sync wherever possible for your future work, for your research work, because it will help you to get free from possibility of hard disk corruption, broken hard disk, or asset lost. It really helps you to keep your data secure, and really helpful. They are very good services. Use trusted freely available software. So trusted means sometimes the software says it's free, but if you see the terms and condition, they may be free for certain number of articles, which is absolutely a waste of time, I believe. Doing taking the software on trial is just for trial. You cannot do it for long term. So just stick to something which is freely available if you if you can't afford the license, or if you think that freely available is really a good enough one for your particular case. So believe on that and just go ahead. Citing original research is strongly recommended rather than an article citing the original article. You need not to cite that reference article. You need to cite the original article for your work. And explore programmatic methods for text mining is also very important aspect. I would say new methods are coming in: knowledge graph, natural language processing of finding the right article, mining the literature, performing quantitative analysis on the articles, what they are saying, doing, making the relationships between the entities are a very good. I believe you should explore those possibilities if you can do hands-on on some of the Python packages, which requires some programmatic skills so that you can really mine good number of articles and do a lot of analysis. So this is all that I have for you in this particular webinar. I must thank you for your patience listening to me all the time here till the end. I hope you find the session interesting and you have learned something new, useful for your research. Best wishes for your scientific journey. Thank you very much.
Thank you so much, sir. So we do have a few queries in the chat. So the first query is from Khadija. She is asking, between two more articles, there are many citations. Should we read those in details too?
So is it like two articles have a lot of variation in the number of citations? Is that the question?
I'll repeat the question once more. Between two more articles, there are many citations. Should we read those in details too?
I would say start with the abstract. Wherever the articles has the has the content which is relevant for your work, there should not be any hesitation to look at the other parameter. If it is if this is covering your topic of interest, just go and read them.
Thank you, sir. And we have another query from Mahanaz. Is this program designed specifically for research scientists?
This particular session, I believe for the other upcoming sessions, they are holistically covering right from the beginning to the some level of advance. We understand that the attendees are the participants are from the varied background. They have some basic understanding, maybe no understanding, or they may be having some intermediate understanding. But there are certain questions they always have in mind and they may be common. So in short, to answer your questions, it is covering right from the basic to some level of intermediate level. And for undergrads, grads, graduates, and postgraduates, and for PhDs as well.
All right, thank you so much, sir. So now I'd like to pass on the stage to Shubham, who will be covering the program registration and cost. So over to you, Shubham.
Thank you, Shigari. So, hello everyone. I'm Shubham. I'm the Business Development and Marketing Associate here at Pan Biotech. So, I'll be briefing you about the registration process for the program, Cheminformatics. Just a second, I'm sharing my screen. Yeah.
Yes, Shubham, we can see your screen.
Yeah, all right. So yeah, as you can see, this is the program page for the like we have program Cheminformatics for Biomedical Drug Discovery. So I'll share you the link of the page so that you can go after the session. So what all you will basically learn from this session is something like 3D structures, then QSAR, docking, and like the molecular dynamics. Now, as you will scroll down, you can see that there is a program overview section. In this, you will get a like summary of the program, like what all the topics you will and concepts you will be learning, like the high throughput screening part, then the docking, as well as how you can use these topics for various research projects if you are from a biotech background or a pharmaceutical background. Along with the, you will get a complete mentor guidance program also, like mentor guidance also. So as you'll scroll down a bit more, you can see that we have a interactive calendar where some of the dates are highlighted like 26, 28, and 29. So today, and on the right side, you can see that we have like what all topics we will be covering in those particular dates. So today on 26 Jan, we had the hands-on workshop part, and your next session will be day after tomorrow, that is on 28th, and you will be learning about the computational structure biology, some of the topics like protein structures and all, some databases also. So yes, now like coming down more, you will see that we have three different tenures for this program. One is 45 days, 60 days, and 90 days. So the resources for this program, the common resources for all the three tenures is you will get the online tutorial, like the complete access of the Omics Logic Learn platform, where you will get the tutorial part and as well as the project examples also, and you will get the coding sessions, hands-on coding sessions also will be there. And by chance if you miss any of these sessions, then we will provide you with the session recording also. Then online mentor and technical support will be provided, and of course after the completion you will get a certificate. Now the difference in these tenure is, like you can see, for the 60 days tenure you have three extra things, or the three extra like opportunities. That is, you will get a one-on-one mentor session, and you can draft your research proposal also. And after that you can like showcase your research proposal or the your research project work as in a poster form with a poster presentation in various international symposiums. And for the 90 days along with the mentor session, you will get an ample amount of time to work on your research project and get it published in the peer reviewed journal. So this is the golden opportunity for the 90 days part. Now coming to the pricing part, as you can see, like the pricing we have mentioned is in USD, and for 45 days it is $150, and for 60 days it is 300, and for the 90 days it is $450. So like if someone is from India, I'll show you in that INR also. So you can see that there is a drop down above. So you just simply have to click on that drop down and select the currency based on your location. So in INR you will find that for 45 days it is 7500, and for 60 days it is 9999, and for 90 days it is 14999. And for African communities, they can also view the amount like in naira. So here are the pricings for our African community. Now how to proceed with the enrollment part or the registration part. So let's say I'm interested for the 90 days part. So I just simply have to click on buy now, and yeah, it is loading. Yeah, you will see this checkout page. Then you can fill up your user details and select the checkbox, and you can select the payment method. You can go for the Stripe or with the PayPal method. And if you like, we have alternative methods also, like bank transfer, credit card, or debit card. So you can opt for that one also. So this all was for the registration part. So over to you, Shugari. Or if you have any queries, please feel free to. Yes, thank you, Shigori.
All right.
Yeah,
Thank you, sir.
and for
Yes, Shubham. Please continue.
yeah, I was just like telling you about to share the marketing at like the contact details, which you have
Okay,
already done.
perfect. So I've got a query from Ankita. She's asking, if I take monthly subscription, may I get access to all the courses of Omics Logic along with certifications?
Yeah, of course. Like if you will, and like for monthly subscription we have, and in that you will get the complete access of the Learn portal. But for the mentor guidance and one-on-one session, you have to opt for the tenures which we have, like for 60 or for the 90 days. But yes, if you want to enroll for the monthly subscription, you can do that. So you can like mail, like just message me your email ID. We can like be in contact with you for the monthly part, subscription part.
She has shared her email ID with me, so we'll be reaching out. So this
All
is
right,
just
then we'll contact.
yes. This is just a note for the participants. Those who are interested in receiving the webinar recording insights and details for enrolling in the program, kindly share your email ID so that we can share the details regarding program curriculum and scholarship options that we have for the participants. And in the chat box, I've also shared the email ID and the contact number of our marketing manager, Sparsthar. So along the way, if the participants are having any queries, you can reach out to us at any point of time. So does anyone have any further queries? If so, please let us know in the chat. And if anyone is left to share their email IDs, please share that as well. Thank you, Fideo. We'll be emailing out to you. Okay, I'll pause for a couple of seconds so that the participants who are yet to share the email IDs, they can share it and be ending the meeting. Thank you so much, everyone.
Thank you, everyone.
Hello.
Yes, Gazara.
Yeah, ma'am. Actually, I had to ask that if we have some specific project that we want to do and be mentored by the mentor at your end, so is it possible to do that?
Dr. Kacker and Shubham, could you please take up that query?
Yeah, like if you have your own particular research question, then definitely you can discuss with the mentor and you can proceed with the research part.
Okay, thank you so much. So whom shall I contact or what email ID I shall contact for this?
You can like contact us at marketingeconomicslogic.com. Then we will connect with you with one of our project manager or with the mentor.
Okay, sure. Thank you so much.
Thank you so much, Shubham. Anyone is left to share? Please share your email IDs and I'll be ending the meeting. All right, I guess everyone has shared. So with that, thank you everyone for joining today's webinar, and we hope to see you all for the program session 1 as well. So thank you everyone. Have a great day.